Thank you so much, and I'll promise I'll be brief so that you all can get to lunch. But my talk is the genomic and serologic evaluation of Alzheimer's disease risk factors in the etiology of glioblastoma. We do have a couple disclosures. I do work full-time as a senior research advocate with Oracle Incorporated, in addition to being a full-time PhD student. And Dr. Kyle Walsh is employed by the federal government as a senior scientific advisor to U.S. Senator Vance in Ohio. So to give you a little bit of background on what is a glioblastoma. So a glioblastoma is the fast-growing, aggressive grade 4 astrocytoma. It's generally localized to just the brain, meaning it doesn't typically metastasize. Due to the genetic composition of GBM tumors, they're inherently resistant to conventional therapies, forcing patients to rely on the gold standard therapies, also called a STUP protocol. This includes surgical resection, radiosurgery, and a commonly used chemotherapy called temozolomide. GBMs have a median survival of around 15 months and a 5-year survival of around 5% after initial diagnosis. Like all brain tumors, glioblastomas are rare, but the incidence of glioblastoma in the population is around 3.21 per 100,000 people. If we take a look at factors associated with etiology of glioblastoma and Alzheimer's disease, glioblastoma and Alzheimer's disease are two neurological conditions with divergent epidemiological risk factors. Of note, several GBM risk factors have been shown to protect against the development of Alzheimer's disease, including male sex, European ancestry, longer telomere length, varicella seronegativity, and higher educational attainment. To further evaluate the etiological heterogeneity between these two diseases, we explored genomic and serum biomarker profiles in patients with GBM, Alzheimer's disease, and neurologically healthy controls. Based on the figure above, you'll notice that the GBM risk is not a monotonic curve. We see that the median age of diagnosis of a GBM is right around the 68 to 70-year-old mark. When you contrast that with Alzheimer's disease, as you all know, the median age of diagnosis is much higher at around 75. Some researchers have made the assumption that given a younger cohort of individuals being diagnosed with GBM and the median survival from date of diagnosis being right around 15 months, that the ongoing progression of the disease is what inherently protects against Alzheimer's disease. However, we'll show in this analysis that we've looked at genomic correlations between both of those two diseases. So in this analysis, we conducted a LD score regression, which is a reliable and efficient method of using genome-wide associated data, or GWAS, summary statistics to evaluate the SNP heritability of complex traits in disease partition into functional categories and ultimately estimate the genetic correlations between different phenotypes. Because this method relies on summary level data and summary level results, LD score regression is computationally controlled even in very large data sets. In this particular analysis, we utilized the 1000 Genome Project European Ancestry sample as a reference. The reason we specifically utilized the European Ancestry sample is that most GWAS analyses to date have been primarily in the European Ancestry analysis, as well as glioma incidence is highest in non-Hispanic whites based on all of the analyses to date. Glioblastoma genotyping data were derived from the most recent meta-analyses of GWAS in glioblastoma. We can see that there were 6,183 cases and 18,169 controls from 9 index studies of individuals from European descent. We obtained the Alzheimer's disease summary statistics from the International Genomics of Alzheimer's Project, or GLIOGENS. And you can see based on the table that we have a twofold increase in the number of cases and controls of GBM compared to Alzheimer's disease. When we conducted the LD score regression, we found that the two diseases were not statistically significant and also were not correlated. However, we are conducting further analyses to truly validate that assumption. Further analyses using Mendelian randomization were also conducted to assess a causal relationship between the data. Does having an Alzheimer's-associated single nucleotide polymorphism, or SNP, have any impact on the risk of a person developing a glioblastoma? However, we found a non-significant protective effect associated with an Alzheimer's disease SNPs. This means having a SNP with a known association to Alzheimer's disease may protect a person against may not protect a person against a glioblastoma. We also did this using a Mendelian randomization to assess whether glioblastoma was associated or whether glioblastoma-associated SNPs had any effect on the risk of Alzheimer's disease. We did observe a protective effect against Alzheimer's disease with 3 highly correlated or associated SNPs. The first, EGFR, the second, TERT, and the last, CDKN2B. This is important because, as mentioned previously, this isn't surprising. We know that longer telomere length is associated with a higher risk of GBM, but also protective against Alzheimer's disease. It's important to take a look at these particular SNPs to better understand possible options for further drug discovery and drug development. But more importantly, is examining the association with amyloid beta serology on GBM and Alzheimer's disease. As all of you know to date, beta amyloid is a hallmark of Alzheimer's disease neuropathology. There are multiple isoforms of amyloid beta that exist. In Alzheimer's disease patients, the ratio of amyloid beta 42 to 40 is decreased in Alzheimer's plasma and can be used as a predictive biomarker in subsequent Alzheimer's disease development in non-diagnosed patients. As in common in most cancer tumor types, there's a buildup of beta amyloid. GBM is no exception to that. Patients with newly diagnosed MGMT unmethylated GBM show a higher ratio of amyloid beta 42 compared to 40 when compared to cognitively controlled patients from the Murdoch study. As a side note, the Murdoch study has been an incredibly long longitudinal study with over 12,000 patients included in that analysis. This higher ratio of amyloid beta 42 to 40 suggests that there is an association with an innate resilience to Alzheimer's like dementias. This increase in ratio of amyloid beta in the bloodstream in gliomas, or in this case, glioblastomas, is aided by gliomas being extensively infiltrated by platelets. Aggressive amyloid and amyloid intermediates are an obligatory part of a tumor environment such as a glioblastoma. So in conclusion, the etiology of GBM and Alzheimer's disease are not just unique, but are directly opposed. This study shows that the examination of other Alzheimer's disease hallmarks, such as p-tau, as we've heard earlier today, have yet not been examined, but are certainly something that's important to consider in further analyses downstream. We hypothesize that there are underlying biological mechanisms at play here, including but not limited to pro versus anti-inflammatory CNS processes. We also suspect that there is possible predisposed changes to neuronal and synaptic architectures, and even possibly polygenetic scores for AD risk being associated with elevated stromal, not immune factors in bulk GBM sample undergoing RNA-seq. We hope to further analyze the utilization of LD score regression and understanding genome correlations between Alzheimer's disease and GBM. I do also want to say thank you so much to Dr. Kyle Walsh for inviting me into his lab as a first-year pharmacoepidemiology student at UNC, and look forward to seeing what we continue to do together, but also want to acknowledge our other partners within Duke, UNC, Stanford, and Mayo Clinic. And I will stop there and say, what questions do you all have?
Yes, the correlation between higher education level and increased GBM risk is interesting. Most diseases show the opposite. Do you have any thoughts on why higher education or greater educational attainment is a risk factor for GBMs?
That's a really interesting question, and I can honestly say that is certainly something we are currently in the process of exploring. Particularly in GBM, we see almost the opposite of many things, but IQ is certainly one of those, and so more to come.
I had kind of the same thought, and I'm wondering what your thoughts have been about the potential for this to signal something about heterogeneity between individuals in either the biology of astrocytes in general or the biology of the glymphatic system.
Ooh, that's a really interesting question as well.
I mean, assuming tumor stem cells of GBM actually are in that lineage, which I don't know how controversial that still is, but
I don't think it really is that controversial to date, but I think it's certainly something that's still being explored. I think there's a lot of unknowns with GBM, in particular the educational attainment, but I can tell you other members of our labs in particular have looked at innate immunity as well as other factors such as autoimmunity in association with an increased risk in that GBM. Some of that includes diabetes, allergies, et cetera, that may also play into the factors that you're asking about as well. Yeah.
Is it possible that higher education leads to stress, specifically in circuits that are modified by astrocyte function? That, I mean, is that a line of investigation that makes sense? That we all understand what the stresses of higher education look like, and feel like. What's that doing to astrocyte function and stem cell function in different areas of the brain? Any thoughts?
Yeah, I think that's certainly what we're in the process of exploring, is taking a look at stress and inflammatory markers associated with that. And I think education falls into that, as we're all here on Friday. But again, I think more to come. I think the interesting thing with this particular project examining Alzheimer's and GBM is that Alzheimer's disease has certainly been something that is incredibly interested and really well funded, and GBM is starting to move up that list. And so I think a lot more discoveries will come taking a look at factors associated with it and increased risk. However, I would say I suspect it's probably related to inflammatory markers as well.